Bismillahirrahmanirrahim. This is the A-Level Biology 9700 syllabus, June 21, paper 2-1. Starting with question 1, figure 1.1 is a transmission electron micrograph of cells from duckweed. I usually don't read this because it unnerves me. Now you can look at it as a diagram and you can see these are whole cells. Now you can see this is the cell, this is the cell, this is really big, this cell is right up to here, here, this is the cell. So you can say transmission duckweed, so some sort of a plant, and then you can see the magnification is 4275. So look at it carefully, understand the question, and then proceed as it is. Calculate the actual width of the cell labeled X. So this width we have to measure this part. So we're going to measure this part, and this is say 1.6 centimeter. So calculate the actual width of the cell labeled X, write down the formula, and you will use to make your calculation and show your working and give your answer in micrometers and 1 decimal place. So be always very careful when you're reading the question. Sometimes you just forget to read this part and you read it, but yet you do not read. Something goes wrong, you're not registering it. So formula, now formula was very easy. A is equal to I over M. Image divided by magnification, and this is the actual, and this is a magnification. So it was very easy. 1.6 centimeter is equal to 16 millimeter. This is equal to 16,000 micrometer, and so you divide it by 4275. So when you divide it by 4275, well, the answer you get is 3.7. And because it said 1 decimal place, so you write here 3.7 micrometer. Micrometer is already written, so you didn't have to write that. Now coming to the next question, P part 1, table 1.1 lists some biological molecules found in plant cells. Complete table 1.1 by choosing one letter from figure 1.1 that indicates a cell structure where each biological molecule is found. And the biological molecules are DNA, cellulose, phospholipid, and histone proteins. Now the important thing is, where would DNA be present? DNA, now let's again look at the cell. Now we look at this picture again. Now let's look at the labels. What are A, B, and C? Now if you look at A is, this is the cell, and this outer boundary, this is the nuclear membrane. You can see the nuclear membrane here, you can see the nuclear membrane here inside this, and this is the nucleolus inside it. And these structures which you are seeing are the chloroplasts. These are the chloroplasts that you're seeing here, and lots in abundance you're seeing them. Now you see this shaded thing which you were supposed to take it out was, this was the nucleus. This was the nucleus. This was the nucleus here. Look at it very carefully. And inside this was the nucleolus. Because you remember photomicrographs is in your syllabus. So you must go through a lot of photomicrographs and electron micrographs. Just Google it and see a lot of micrographs and see how they are different types, which you should be able to label anything in it. So where was DNA present? DNA was present in A. DNA was present in B, which is inside the nucleolus. And DNA was present in C. Now what is C? C is the chloroplast. You know there is a loop of DNA in the chloroplast. So DNA was present in A, B, or C. So you could have said DNA A, you could have said B, you could have said C. All three were correct. You don't have to write all three of them. You only have to write one of them. Then the next thing was cellulose. Now cellulose, as you can see, is E. E is the cellulose cell wall. Where is it being labeled? This is being labeled here. So this is the cell wall. So E, cell wall, was cellulose was present in E. Then we have phospholipid. So where was phospholipid present? Phospholipid was present in A and C. Why A and C? A is the nuclear membrane. So phospholipid was present here. And why C? What is C? C is also the chloroplast. In the chloroplast also the outer membrane and the inner membrane and the thylakoids are all made up of phospholipids. So then we had to say phospholipid was in A and C. Any of these was correct. Then we come to the last one, histone proteins. Now where were histone proteins present? Now histone proteins are present only in the nucleus. So histone proteins would either be in A or it would be in B. So because remember, in the loop of DNA which is present in the chloroplast or the mitochondria does not have any histone proteins. And that's a very important point you need to remember. We might ask you that in the MCQ. So histone proteins were present in A or present in B. So you have to write one of them, but please understand why these were the answers to it. Then it says state the name of a cell structure visible in figure 1.1 where ATP is synthesized. Now state the name of a cell structure visible in figure where ATP is synthesized. Now that could only be a mitochondrion or a chloroplast. Now that's the syllabus point. But you all remember the mitochondrion part, but you forget the chloroplast part. Please remember in chloroplast also, if somebody is in A2, they would know that photophosphorylation takes place. While in the mitochondria, you know, of course, there's substrate level phosphorylation and oxidative phosphorylation. So mitochondria also ATP is synthesized, and in the chloroplast also ATP is synthesized. This is in the AS level. Name a cell structure that produces RNA. Well, where will RNA be produced? Wherever there is what? There has to be DNA and there has to be ribosome. So these two things will be present. If DNA and ribosomes are present, only then mRNA can be made. So where can mRNA be made? mRNA can be made only if you require it. It's only going to be the nucleus. But then, as an addition has been made in the mark scheme, it can also be chloroplast. It can also be mitochondrion. Because as I said earlier, wherever there is DNA and wherever there are ribosomes, that's where transcription can take place. So they could have also asked you name a cell structure that produces mRNA, name a cell structure where transcription takes place. Then it says number C part, describe the evidence from figure 1.1 that shows that the image is a transmission electron micrograph. You know, we do two electron micrographs, scanning and transmission electron micrographs. That's the syllabus point. If you go through the syllabus point wise, I'm sure you can all understand it and do this very well. But if you don't go through syllabus, then you're just going to read from the book or notes. I'm sorry, you're not going to achieve much. Now, the main thing you've got to remember is that electron micrographs has a very higher magnification. They have a very, very high magnification. The maximum useful magnification in a light microscope is X1500. I said maximum useful magnification. You can magnify it, but it's not used. It just gets blurred. Just like you zoom a picture, it gets blurred. It doesn't get very clear. You can't see the details of it. When you say better quality of a picture means that you see more details of it. You don't say it becomes more clear. That's all rubbish. That's wrong biological English. So describe the evidence that shows the image is a transmission electron microscope is a higher resolution. Number two, internal structures of the chloroplast can be seen, which cannot be seen under higher magnification under the light microscope. It's a very higher magnification, more than 1000, because it was 4275, and it's a very thin slice. And number two, you do not see any surface features, because if it was a scanning electron micrograph, you would see the surface features of it. And this is not giving you any 3D image of it. So no surface features are shown. So that is why it's a transmission. Transmission means it passes through and through the structure. So you transmits. So it passes through it, so you can see the details of it. Like for instance, when you're flying, I mean, I've just been abroad in an aeroplane. I was coming back, and when I was landing in Lahore, I could see the rooftops of all the houses. But I couldn't see the details of the inside of the houses. How many rooms were in the house? How many doors and windows were in the house? I could just see the surface. I could just see the rooftops of all the buildings. But if you have that's a scanning electron micrograph. But if you see a transmission means that it passes through and through it, and you can see the details of the internal structures of the cell. Now coming to question number 2, figure 2.1 shows 3 molecules of water. Discuss the hydrogen bonding that occurs between the water molecules shown in figure 2.1. So describe the hydrogen bonding, not discuss, describe the hydrogen bonding. So describe the hydrogen bonding means that if you look at the diagram shows hydrogen bond is a weak bond. Each oxygen atom forms 2 hydrogen bonds. Each oxygen atom forms 2 hydrogen bonds. These are the 2 hydrogen bonds. Then between oxygen atoms of one water molecule and hydrogen of another. Then water is dipolar. Okay, then if you give a little more details are oxygen has a small negative charge and hydrogen has a small positive charge. Dipole means what? Dipole means that Electrons are not shared equally between oxygen and hydrogen. So basically you had to give me all that, and this description would get you your 3 marks. So you had to give me 3 points and you'd get your 3 marks. Now basically main thing is you've got to learn to word this. Some of these you'll have to sort of memorize. Hydrogen bond is a weak bond. Each oxygen forms 2 hydrogen bonds between oxygen of one water molecule and hydrogen of another. Water is a dipole. Oxygen has a small negative charge and a partial negative and a partial positive charge on the hydrogen. So that wording all this, maybe you know it, but if you don't know how to word it, I'm sorry, it's not going to help you. You've got to learn the exact wordings which have been used in the mark schemes, and this is what you need to understand. The human enzyme salivary amylase is composed of one polypeptide. Figure 2.2 represents the structure of a molecule of salivary amylose. If one polypeptide, that means there's no quaternary structure. Because the quaternary structure will only come into effect if there are 2 or more polypeptide chains. So you've got to understand, they've told you this in the question, one polypeptide. Now as you go ahead, it says explain the role of hydrogen bonding in maintaining the secondary structure of proteins, such as salivary amylase. Now basically the secondary structure is made up of hydrogen bonds, but what are they? They are the alpha helices and the beta pleated sheet. There's only one mark for it. So the hydrogen bonds maintain the shape, or you can form the alpha helices and the beta pleated sheet. So alpha helices all allow the formation of alpha helices and beta pleated sheets. So basically it's the maintaining the shape of it, and that is because of that. Then coming to the part 2, explain the role of hydrogen bonding in maintaining the tertiary structure of proteins such as salivary amylase. Now tertiary structure is what? Tertiary structure is going to further stabilize it. So hydrogen bonds help to stabilize it. And then between the R groups with amine and carboxyl groups, and maybe between amino acids far apart in the primary structure. And this will help to maintain the globular shape, and of course will maintain the shape of the active site because it's an enzyme. So the active site is the most important part of an enzyme. So the wording of it, hydrogen bonds help to stabilize between R groups with amine and carboxyl groups, maybe between amino acids far apart in the primary structure, and helps to maintain the shape of the active site. Then coming to the C part of the question, outline the importance of water as a solvent in plants. Now there's a very basic question. Why do we need water in plants? Well, basically they dissolve all the ions, the minerals or the salts or the polar molecules. You can use any of those words. Then they transport solutes in the xylem and the phloem. Then the storage of solutes in the vacuoles. Your vacuoles is mostly water, the large central vacuoles, all water. So it stores solutes in the vacuoles. Then chemical reactions occur in water. You see how we need hydrolysis and condensation. Everywhere we talk of that. Starch to glucose, glucose to starch. And then dissolves carbon dioxide and oxygen in connection with, you can talk about respiration and photosynthesis. So all these points help it tells you the importance of water as a solvent in plants. So just giving it to you in words, please pause the video here and learn these marking scheme points. So dissolves ions and polar molecules, transport solutes in xylem and phloem, storage of solutes in vacuoles. You see, vacuoles are made up of water, as I just said. And chemical reactions occur in water. Dissolves carbon dioxide in the process of photosynthesis, or you can say dissolves oxygen for respiration. So this is all one mark scheme point. This is an OR. Now coming to question number 3, whisking tubing can be used to investigate the properties of cell membranes. A student carried out an experiment to investigate osmosis using whisking tubing. An outline of the investigation is shown in figure 3.1. So let's look at it very carefully. There is 15 centimeter of sucrose solution, 0.9 mole dm cube in this, and then we have the whisking tubing which has been labeled this. So this is the whisking tubing. I'm just going to use a color for it. This is the whisking tubing, and this is a knot at it. We've tied a knot at it. So this is the same whisking tubing here, and there's fluid in it, and there's this knot here, and then there's the fluid here, and then there's a knot here. And then please everybody look at the level. This was the level here, this was the level here, and now this is the level here. So whisking tubing, meniscus, sucrose solution. Inside it we have the sucrose solution, which let's shade it yellow. So we've got the sucrose solution here, tap, we've got the top of the knot in the whisking tubing, and then we have 15 centimeter cube of sucrose solution of 0.9 mole dm cube. So look at it very carefully, and then there's a ruler behind it. There's a measuring ruler, say centimeter, millimeter. 6 pieces of whisking tubing were filled with 10 centimeter cube of different concentrations of sucrose. One had 0.0, one had 0.4, 0.8, 1.2, 1.6, and 2.0. So we have how many? We have six. 1, 2, 3, 4, 5, and 6. So we have six concentrations of different concentrations of sucrose. The height of the meniscus of each solution in the whisking tubing was measured. The pieces of whisking tube were put into test tubes containing 15 centimeter cube of 0.9. So 0.9 was the standard which was outside. Inside in one we had 0, outside 0, 0.9, 0.4, 0.9, 0.8, 0.9, 1.2, 0.9 and 2.0, 0.9. So outside it was always 0.9. The pieces of whisking tube were put into test tubes containing 15 cm cube. So fluid was 15 cm cube of 0.9 mole dm cube sucrose solution. After 20 minutes, the pieces of whisking tubing were removed from the test tubes and the height of the meniscus in each was measured. So we measured the height of this. This was the height that we measured, right? The results are shown in table 3.1. Now let's look at the results. So concentration of sucrose 0, we had minus 12 distance in height over meniscus after height of meniscus after 20 minutes. Minus 12, then 0.4 minus 4, then minus 2, and then plus 1, plus 6, and plus 11. Now when you look at part A, the whisking tubing used by the student was not permeable to sucrose. Explain the result shown in table 3.1. So you have to explain this result. What happened when the whisking tubing had 0 inside and you had 0.9 outside? Why did it go to minus 12? The reason why it went to minus 12 is because all the water moved out, so it went down. Then it was what? Then it was inside 0.4. Outside it was still 0.9. Even then it is going to move out. Then it was 0.8 inside and outside was 0.9. So again it will move out. But then it was what? Then it was 1.2 inside. It was 1.2 inside. And outside was 0.9. So then it's going to move in. So it's going to go up plus 1. Then it was 1.6 inside. And 0.9 outside. Even then from it's going to go from here to here. Then it was 2.0. Then it was 2.0 inside. And outside was 0.9. So you know from 0.9 it's going to go inside. It's going to go plus 11. So how will you explain this? Now it says the whisking tube and there's 3 marks. You have to explain this and it's not permeable to sucrose. So basically water was moving in and that is what you had to give me. That is what you had to tell me is that concentration of sucrose 0, 0.5, 0.8 was less than 0.9. So water moves out of the whisking tubing. And then concentration of sucrose 1.2, 1.6 and 2 were more than 0.9. So water moves into the whisking tubing. And the fact there is, you had to refer to the net movement of water. So there was net water movement. Either the net water movement was outside or inside. So external solution at times had a higher water potential than the whisking tubing. So water will move down the water potential gradient. So all you had to give me was all this information, but in the right biological English. The concentration of sucrose 0, 0.4, 0.8 are less than 0.9. So water moves out of whisking tubing. This was the first mark scheme point. Then concentration of sucrose 1.2, 1.6 and 2.0 is more than 0.9. So water moves into the whisking tubing. You have to talk about the net movement of water. That's a very important word, net movement of water. So whenever it's occurring, whichever direction it's occurring, that means the net movement of water is more at that time. Net movement of water either into the whisking tubing or out of the whisking tubing. So net movement of water is a very important word that you must use, and whenever there's a question on such sort of a question. When external solution has a higher water potential, then water moves down the water potential gradient. So basically you have to use the word water potential gradient. The fact you have to use the word higher water potential. When is there a higher water potential? Now these are all biological terms and only we can give it if you know about it. Coming to the B part of the question. When red blood cells are placed in water, they're destroyed by bursting. The student also investigated how red blood cells are affected by immersion in solutions of sodium chloride of different concentrations. Blood samples of the same volume were added to solutions of sodium chloride in separate test tubes. After 10 minutes, the student took 0.1 cm³ of the blood sample from the test tube and estimated the percentage of red blood cells that had burst in each blood sample. Percentage. Didn't say 2 cells have burst, didn't say 5 cells. So must have measured say 20, 30, 40 and then seen how many of them. So for discuss estimated the percentage of red blood cells, 10%, 20%, 30%. Figure 3.2 shows the student's results. Percentage of red blood cells destroyed by bursting. So you can see here now there is a straight line. Here from here to here is a straight line. So between these values you can see 100% had burst. And then we are saying the percentage is decreasing from here till here. So till here to here you say decreasing, and then it is percentage of red blood cells, there is no bursting from here to here. So none of them have burst here to here. 0%. So there's 0%. So look at the graph, look at it carefully, do what I'm doing is telling you to do. So you have the concentration of sodium chloride. So between 0 and 0.04 is somewhere here. 0.04 there is 100% have burst, and then at 4 it is decreasing. So you look at that, and let's look at the next question. The question is describe and explain. When it says describe and explain, so description is something else. It's no biology. Describe means no biology. Explain means you have to give me some sort of a biological explanation. So describe and explain the effects on red blood cells of immersion in different concentrations of sodium chloride. Now the describe mark, though there are 4 marks for this, and you give me say 6 explanations and they're all correct, but I can only give you 3 marks because there's 1 mark for the describe. There is 1 mark for the describe and there's 3 marks for the explain. So even if you give me all the explanation points, even maybe more, I will only give you 3 out of 4 because there's 1 mark for the describe. Now if you look at all these describe, these are all the describe. Please remember I'm giving you all the describe first. At concentration less than 0.04, all cells burst. At concentration between 0.04 and 0.14, decreasing percentage of cells burst. This is no biology, it's just what you could see from the graph. There's nothing else. You just have to look at the graph and you have to give me these figures. So at concentration between 0.04 and 0.14, decreasing percentage of cells burst. But I will still get no marks for this statement because what have I forgotten? I've forgotten to write the units. So if I didn't write the units, I'm sorry, I'm not going to get any marks. So this describe, I haven't written here also the units, here also I haven't written the units. So here I've written the units. So at concentration greater than 0.14 mole dm cube, no cells burst. So you have to write the units. You have to give me the units because it is a describe. You have to give me whatever the units were given here in the graph. So please remember that just by forgetting to write the units, you will lose a mark. So this was all the describe. Any one of these would get you your 1 for the describe. Now let's go on to the explanation. Now if you look at all the different points for explanations, there are a whole lot of them. There are about 5 or 6 of them. And in low concentration, water moves into cells down the water potential gradient. Cells increase in volume. The RBCs burst as they have no cell wall. Or you could have said cell membranes are not strong enough to withstand the increase in volume or pressure. Then between 0.04 and 0.14 mole dm cube, water potential gradient into cell decreases. Above 0.14, water potential inside the cell is the same. At high concentration, more than 0.14, water leaves the cells and the cells shrink or the cells show crenation. It's not cremation. Cremation is when you burn the dead bodies. Crenation, it's an N. Don't write cremation, please. That means something else. So crenation. So at high concentration, water leaves the cell, cells shrink, cells shrivel up, or cells show crenation. And so this is the explanation. But you haven't to give me all these. You have to give me any 3 of these, and you would have got your full marks for this question. So that I'm going to finish the video here. I'm going to continue the rest of the 3 questions in the second video. And please do subscribe. And please do leave your comments if there's anything else which I can do to help you all out in the last few days left before the exams. All the best and thank you very much.